there are the drinkers. We're learning about this form of addiction from a test animal researchers call bar flies.
The directions for a human being are written in code, 3 billion letters long. These instructions tell our bodies how to live, how to grow, how to die. Researchers call this code the sequence.
Moving right along from one addiction to another. It's a familiar scene in bars and parties. One person consumes one drink after another and never appears to get drunk, while someone else barely finishes off a single glass and is instantly tipsy. How come certain people can hold their liquor better than others? And why do some people become addicted to it?
Barely moving.
That's good.
And these flies are running
Ulrike Haberlein spends her days with bar flies and cheap dates, but she's not hanging out at the corner pub. She's studying the genetic roots of alcoholism. Her primary tools, millions of tiny fruit flies, alcohol, and the inebriameter, a device that, as its name implies, gets flies drunk.
We have fun naming our flies. So we have cheap date, we have lightweight, we have tipsy, we have barfly, we have hangover. The cheap date flies get intoxicated quite easily. So do tipsy flies, and so do the lightweight flies. The bar flies are intoxicated at much higher alcohol doses, so they resist alcohol much better.
Haberlein and her team studied these varieties of flies and many others in an effort to identify genes that regulate the effects of alcohol.
Alcohol induces a change in behavior such as it stimulates the flies. They run around more rapidly. What you can do then is go into the fly genome and mutate one gene at a time in a completely random fashion, and then ask how does that mutation affect the behavior that you're interested in.
Haberlein has identified a gene that controls a fly's sensitivity to alcohol. Normally, this gene produces a signal that triggers changes in the messenger chemicals that tell a cell how to respond to its environment. When alcohol invades the cell, the gene tells it to act differently. A chain reaction goes off throughout the body, eventually affecting things like memory and coordination. When Haberlein knocked out the gene that controls the chemical triggers, she found that the flies got drunk more easily. The same gene hasn't been identified in humans, but a similar molecular pathway exists. After all, more than 60% of the human genes that have been identified have counterparts in fruit flies. Haberlein says humans are just big flies without wings.
It's dramatic when you look at a fruit fly. It's a tiny, tiny little organism, and you look at us, we don't really look very much alike. So it is, I think, shocking to even some of the people that are educated in the field to find that at the molecular level there's so many similarities. There goes the alcohol. So you can see the immediate effect that alcohol has on the behavior of the flies. Again, these are quite calm, and these flies are running around like maniacs.
Look familiar? The flies' responses to alcohol, so similar to humans, means that some of our molecular pathways may be alike.
As you give them more and more alcohol, then they start becoming quite uncoordinated. You will see them fall over, they'll stand back up, they'll fall over. And then eventually they really just fall on their sides and become immobile and can no longer right themselves.
Haberlein's colleague, Kirk Wilhelmsen, wants to find not just the matching gene in humans, but also what makes some people more susceptible to alcohol and more likely to become addicted.
It turns out that if you look at a gene in a fruit fly, you usually can find a similar gene in humans. And so if she tells me that if I mutate a gene in a fruit fly and it causes a fruit fly to be more sensitive to alcoholism, that encourages me to look to see if I look in that gene, can I find normal variations in that gene among the alcoholics compared to the non-alcoholics to see if the gene plays a similar role in humans.
10 to 15% of Americans develop alcoholism, and the numbers grow within families. There's a 30 to 60% chance that a sibling of an alcoholic will be one too.
They say that the apple doesn't fall far from the tree. Well, it's true for alcoholism. If you look at families that have alcoholics, there's not just one, there tends to be more than one. That doesn't mean that everyone that's in that family will become an alcoholic, but it means that there's something going on there. There's an increased risk.
But genes aren't the only factors that make people alcoholics. Environment plays a part too. In their studies of families, Wilhelmsen and his team analyzed volunteers' blood samples, isolate the DNA, and look at the pattern of inheritance of certain genes.
So these two people are siblings here, and you can see they share one chromosome.
Wilhelmsen has found that there is an inherited component to alcoholism. But he doesn't know yet how many genes are involved, how they interact with each other, or how they are affected by the environment.
It's more likely that there won't be a single gene that actually can predict whether someone becomes an alcoholic or not. There may be a gene, for instance, that contributes to craving. There may be a gene that contributes to the withdrawal symptoms. There may be genes that contribute to antisocial behavior and risk-seeking behavior. And each of those genes may contribute to this larger picture of alcoholism.
Researchers hope to understand the causes of alcoholism, as well as to target treatments more precisely.
We use the flies to identify the genes, but those genes need to be validated in more sophisticated models and eventually in humans. Our big goal is to identify many genes in the fly that regulate these behaviors and provide human geneticists with candidate genes that they can study. So at the end of all of this, if even one of our genes ends up being relevant to human alcoholism, I will feel that all of these years have been worth it.
The Secrets of the Sequence teaching materials were developed at Virginia Commonwealth University with funding from the National Academy of Sciences and the Pfizer Foundation. The original public television series, Secrets of the Sequence, was produced by Ward Television with funding from Pfizer, the Pfizer Foundation, Oracle, and the Council for Biotechnology Information. Special thanks to member institutions of the Series Advisory Board, consisting of Virginia Commonwealth University, Harvard University, University of Wisconsin, University of Michigan, University of California at San Francisco, and the MRC Laboratory of Molecular Biology, Cambridge, England.